We are back. I believe we're now on part 5 of our chapter 5 microscopy demo here. And we were talking about how to use our microscopes. We had discussed how to get our specimen in focus, starting with scanning, moving up to low power and then high dry power. And now we're ready to move up to the oil immersion lens. And here we have to be really careful because the oil immersion lens is so long. We are almost making contact with our glass slide, so we have to be careful not to damage our specimen or our oil immersion lens. So because the oil immersion lens, the diameter is so small, and because we're working so close to our specimen, it's impossible to harvest enough photons of light leaving the specimen, moving through the mirror lens system and then striking our retina. If there's only air in that space between the specimen and the oil immersion lens, through the refraction of light, which is a result of light changing speed as it moves from glass into air, it changes speed, and as a consequence it bends or refracts, and that means we're going to lose photons of light, and consequently we won't have enough photons striking the retina, there won't be enough action potentials generated, our visual center in the brain receives too few data points and struggles to create an outline of the image. And what we will see, what we perceive, is an object that's very fuzzy looking. We can't tell, are there two single cells present? Is there one big bilobe cell present? Because it looks fuzzy and it's really unpleasant. So to reduce light refraction, what we want to do is fill that airspace between the specimen and our oil immersion lens with a substance through which light will travel about the same speed. And that substance that was discovered is a special high-quality oil called, if I can find it, immersion oil. So what we're going to do is we're going to take a drop of immersion oil, place it right on top of our specimen. In this case, there's a cover slip present, but later on we're going to be looking at bacteria that have been stained that don't have a cover slip on them. We're actually going to be putting the immersion oil right on top of those stained bacteria, and then we'll rotate the oil immersion lens into place right into that oil. And as a consequence, then, we'll reduce light refraction and increase resolution. Now, the process would be, I would have gotten my specimen in focus using scanning, then low power, then high dry, right? So we're presuming we've done that. So now I'm going to rotate my high dry lens out of place, and I'm going to get my immersion oil, and I'm going to place a drop of immersion oil right on top of where the light is striking my microscope specimen. Okay. And then very slowly and gently, looking from the side, I'm going to rotate my oil immersion lens into place. And if I've done this correctly, you can see that the oil immersion lens is immersed. It's touching the oil. We don't want it touching the slide, but we want the oil immersion lens immersed, touching the immersion oil. So now as light that strikes the microbe and is moving upward, now as it moves from glass into the immersion oil, it's not going to change speed as dramatically. It will reduce refraction, it won't bend as much, more of the photons will flow straight into the oil immersion lens, and consequently we're going to harvest more photons striking the retina, more action potentials, more data points for the visual center of the brain that then can create this nice clear outline of cells, thus increasing resolution. So we use immersion oil to decrease light refraction. The consequence is we're going to increase resolution. Okay, now do remember, folks, with using the immersion oil, which focus adjustment knob can we use? And remember, we can only use that inner fine focus adjustment knob. And again, it should only take 1 to 2 turns maximum to get your specimen into focus. If it's taking more than that, again, ask one of your professors or your wonderful micro tutors to give you a hand. Okay, I'm going to stop here.